Hello students, I am teacher Svetlana and in our today's lecture we will learn further the biomolecules in a cell, nucleic acids. We have already seen the DNA, today we will see the RNA and the enzymes with respect to its nature, properties, nomenclature and classification which is covered in the chapter Biomolecules. So let's begin. Students, another nucleic acid that is found in living organism is the ribose nucleic acid, which is also called as RNA. In most of the organisms, it is not found to be hereditary material, but in certain organisms like tobacco mosaic virus, it is the hereditary material. Like the DNA, ribose nucleic acid also consists of polynucleotide chain with the difference that it consists of a single strand. Here, have a look at this image. This is the structure of your RNA, and here for your reference, I have put the structure of the DNA also. So the basic differences in the RNA, you have a single strand, and the DNA, you have double strand. The nucleotides of the RNA have ribose sugar instead of the deoxyribose sugar as in case of DNA. So this is the second difference, that we have ribose sugar in RNA, and on the other hand in the DNA we have the deoxyribose sugar. In case of RNA, uracil substitutes thymine of the DNA. So this is the third difference. Purine pyrimidine equality is not found in RNA molecule because of its single-stranded structure. Obviously, since it's a single-stranded structure, there is no complementary base pairing. So there is no purine pyrimidine equality found. RNA strand is usually found folded upon itself in certain regions or entirely. So it's very rare that entirely it will be folded, but at times the RNA is folded at some regions. This folding helps in stability of the RNA molecule. Most of the RNA polynucleotide chains start either with adenine or guanine. Now let us have a look at the types of the RNA. There are three types of the cellular RNA, which are the messenger RNA, mRNA, or it is also called as template RNA. Then the second type is the ribosomal RNA, also called as rRNA. And the third type is the transfer RNA, which is called as tRNA or soluble RNA. The mRNA, it carries the genetic information for arranging amino acids in definite sequences. It is a linear polynucleotide. It accounts 3% of the cellular RNA. So have a look at this image here. This is your mRNA or messenger RNA. It is linear. Its molecular weight is several millions. mRNA molecule carries information to form a complete polypeptide chain is called as the cistron. So it has the information for synthesis of a polypeptide chain, that is basically synthesis of proteins. Size of the mRNA is related to the size of the message it contains. Synthesis of mRNA begins at 5' end of the DNA strand and terminates at the 3' end. So this is the polarity of your mRNA, 5', 3'. Then we have the rRNA. You can see here how they are used for the synthesis of proteins. They have codons, which is a triplet code, and it codes for a specific amino acid. So these amino acids then come in sequence to form the polypeptide chain. rRNA forms 50 to 60% part of ribosome. It accounts 80 to 90% of the cellular RNA. It is synthesized in the nucleus. Colin in 1960 discovered it. It gets coiled here and there due to intrachain complementary base pairing. You can see this is your ribosomal RNA. In between, it shows a little bit of complementary base pairing. The third type is your tRNA or the transfer RNA molecule. These are much smaller, consisting of 70 to 80 nucleotides. It is also single-stranded, but to number of complementary base sequences after pairing, it is shaped like a cloverleaf. So it takes a cloverleaf shape due to complementary base pairing. Each transfer RNA can pick up a particular amino acids. So basically, all your different types of these RNAs have a very important role to play at the time of protein synthesis. So transfer RNAs have the role of transferring or transporting your amino acids. Following four parts can be recognized on the tRNA. The DHU arm, which is here, and it is also called as the D loop, which is called as dihydroxyuracil loop, or the amino acid recognition site. Then you have the amino acid binding site. This is your at the 3' end, it has an amino acid binding site. Then you have an anticodon loop or the codon recognition site. Here, this is the anticodon loop. Then we have the ribosome recognition site. In the anticodon loop of the tRNA, 3 unpaired nucleotides are present, called as anticodon, which pair with codon present on the mRNA. So here you have the anticodon loops. It pairs with the codons on the mRNA. So whatever is the codon on the mRNA, a complementary codon will be, which is called as the anticodon, is present on the transfer RNA. The specific amino acid is attached at the 3' end in the acceptor stem of the clover leaf of your tRNA. So here you have the amino acid which it carries along with itself at the time of protein synthesis. Now students, let us have a look at the next important macromolecule, which are enzymes. Thousands of different chemical reactions takes place automatically at a given time in a tiny living cell. The reactions taking place at a body temperature. So you have thousands of reactions happening within your cell, and all this happens at your normal body temperature. If these enzymes were not present in the cell, either the reactions would not occur, or if they occurred, they would occur at a very, very slow rate. And hence, we have each enzyme that catalyzes a small number of reactions specifically, or perhaps only one. So you have enzymes which catalyze just a few reactions, or sometimes you have just one enzyme that catalyzes one specific reaction. This substance upon which an enzyme acts is termed as the substrate. So on the substance where the enzyme activity is performed is called as the substrate. The enzyme which acts within the cell in which they are synthesized are known as endoenzymes. Example, enzymes produced in the chloroplast and mitochondria. If they act outside the cell in which they are synthesized, they are known as exoenzymes. Enzymes released by many fungi. These enzymes synthesized by living cells retain their catalytic property even when extracted from the cells. So you have two types of enzymes: endoenzymes, where the enzyme activity is performed in the same cell where it is produced, and you have exoenzymes. These are those enzymes which perform their activity outside the cell in which they are produced. However, even if they are extracted out of the living cells, they still retain their catalytic properties. Nature of these enzymes: on the basis of the chemical composition, enzymes can be put into two categories: purely proteinous enzymes, that is proteases that split proteins, and you have conjugated enzymes. They are made up of proteins to which a non-protein prosthetic group is added. The prosthetic group is firmly bound to the protein component by chemical bond and is not removed by hydrolysis. If the prosthetic group is removed, the protein part of the enzyme becomes inactive. Have a look at this image here. This will show you a schematic representation of the classification of enzymes based on its nature. You have simple enzymes which are made up of just proteins, and then you have conjugated enzymes which are made up of protein and a non-proteinic part. These are enzymes which require certain organic compounds and inorganic ions for their activity. The organic compounds that are tightly attached to the protein part are called as coenzymes, whereas the inorganic ions which are loosely attached to the protein part are called as cofactors. So if you have an organic compound that is associated with the protein, then they are called as coenzymes, and if it is an inorganic ion, then they are called as cofactors. Examples of coenzymes are your NAD, which is called as nicotinamine adenine dinucleotide, and then you have FMN, which is called as flavin mononucleotide. And on the other hand, the examples of cofactors are basically ions of magnesium, copper, zinc, iron, manganese, etc. Properties of enzymes: proteinaceous nature. All enzymes are basically made up of proteins. Then they have three-dimensional conformation. All enzymes have specific three-dimensional conformations. They have one or more active site to which substrate, that is the reactant, combines, and the points of the active site where the substrate joins with the enzyme is called as the substrate binding site. Then it has catalytic property. Enzymes are like inorganic catalysts and influence the speed of the biochemical reactions, but they themselves remain unchanged. So you must have come across these properties of a catalyst in your chemistry. So it's just the same. After completion of the reaction and release of the product, they remain active to catalyze again. A small quantity of the enzyme can catalyze the transformation of a very large quantity of substrate into an end product. So you require very small quantities of enzyme to catalyze a reaction. Specificity of the action: the ability of an enzyme to catalyze one specific reaction, and essentially no other, is perhaps its most significant property. Each enzyme acts upon a specific substrate or a specific group of substrates. So enzymes are very specific. They act only on its own substrate. Reversibility of the action: enzymes are very sensitive to the temperatures and pH. Each enzyme exhibits its highest activity at a specific pH, which is called as the optimum pH. Any increase or decrease in the pH causes a decline in the enzyme activity, and similarly is the effects of temperatures. Enzymes are destroyed at high temperatures of 60 to 70 degrees Celsius or below. They are not destroyed, but they become inactive. This inactive state is temporary, and the enzyme can be active at suitable temperatures. Most of the enzymes work at the optimum temperatures, which is between 20 degrees to 35 degrees Celsius. Nomenclature: there are various ways of naming the enzymes. Enzymes are named by adding the suffix asa, A's, to the name of the substrate on which they act. They are sometimes named according to the type and the functions they perform. Sometimes they are named according to the source from which they are obtained. According to the International Code of Enzyme Nomenclature, the name of the enzyme ends with an A's and consists of double name. The first name indicates the nature of the substrate upon which the enzyme acts, and the second name indicates the reaction catalyzed. Example: pyruvate decarboxylase catalyzes the removal of carbon dioxide from the substrate pyruvic acid. Classification of enzymes: here you have the 6 major classes of enzymes. Oxidoreductases: these are enzymes catalyzing oxidation and reduction reaction by transfer of the hydrogen and or the oxygen. Example: alcohol dehydrogenase. Then you have transferases: these enzymes catalyze transfer of certain group between molecules. Example: glucokinase. Hydrolases: these are enzymes catalyzing hydrolytic reactions. These class include amylases, proteases, lipases, etc. Example: sucrase, lyases. These enzymes are involved in elimination reactions resulting in the removal of a group of atoms from the substrate molecule to leave a double bond. It includes aldolases, decarboxylases, and dehydratases. Example: fumarate hydratase. Isomerases: these enzymes catalyze structural rearrangements within the molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as racemases, epimerases, isomerases, mutases. Example: xylose isomerase. Then you have ligases or synthetases. These are the enzymes which catalyze the covalent linkage of the molecules utilizing the energy obtained from hydrolysis of an energy-rich compound like ATP, GTP. So this basically brings about attachment of two molecules. Example: glucothionate synthetase, pyruvate carboxylase, etc. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.